A warm welcome to the very first seminar series here at the CVJ-Centre at Kochi. It's a real pleasure to have you all with us. This is the first seminar in our series. Please stay tuned for more in the coming months. For those joining from outside of Kochi, our industry-funded Center for Synthetic Biology and Biomanufacturing is on a mission to build a world-class biomanufacturing hub in India, bringing together cutting-edge research, hands-on training, and India's first MTech program in synthetic biology and biomanufacturing in partnership with QSAT, funded by the industry, Synthite. It's a rare honor to introduce Dr. Girinath Pillai, a computational chemist whose work doesn't just advance science on paper, but reaches people in the real world. Excited to share that he designed a therapeutic molecule that's now in clinical trials in UK. As principal scientist here at the CVJ Centre, Dr. Pillai is building a pioneering bio-AI division, harnessing artificial intelligence to determine how the medicines and materials of the future will be made. Prior to joining CVJ Centre, he led a computational drug discovery team at Signature Discovery, one of the Europe's most respected research organizations. Trained at the University of Florida in the US and at the University of Tartu in Estonia, Dr. Pillai's career spans continents and disciplines, united by one conviction that computational intelligence can solve life's hardest problems. Thank you all for joining this primordial step from CVJ Centre. The floor is all yours, Dr. Pillai.
Thank you so much, Dr. Pawan, for this nice introduction and kind words. And thank you also giving a kind introduction about our center. Just want to reconfirm, I hope everybody can see my screen, which tells you the title of this webinar, which is AI Programming for Life. You can use the chat.
Yes.
Yeah. Oh, thank you. Thank you, Dr. Pawan. Just before we start, I posted a link, 2 links in the chat for an icebreaker session. We will discuss about that in a moment. So those who have recently joined, it is there in the chat. If you scroll up, you can see 2 links, and please be ready with that. Thank you. I won't be able to see the chat as I'm sharing my screen. Great. Welcome all. Thank you so much for taking some time from your busy schedule to listen to some of the AI Programming for Life insights. So as an icebreaker session, I want you to please go to aistudio.google.com. You can use your Google account, and please make sure, let me enable the highlighter. Yeah, please make sure you delete or close this option grounding with the Google search. And please use this question, what is today's date? This is a question you have to ask. If someone is getting some interesting answer, you can post it in the chat, please. Likewise, please go to gemini.google.com, which is a common app everybody now uses on their Android phones, and ask the same question, what is today's date? You can put it in your chat message if you see something different. And I'll be starting my slides based on that. I'll just give 1 minute for that, which is very, very important for today's session. So can someone put it in the chat if you got some different answers or same answers, asking the same question? And please don't forget, Don't forget to close this option. Grounding with Google search, you have to close that option and then ask the question. Different answers. Yes, Nived got today's date as May 22, 2025. But in Gemini, you got the actual today's date. And that's what we are going to learn today, why that is different. The same models, the same Google Gemma models, why they are giving two different answers. And this is why we call AI cannot easily replace us. They need us. So I start my webinar with saying that AI can ever replace humans. They need humans to move forward. Now let's see why the responses are different. Gemini is mainly for everyday consumers, and AI Studio is for developers and proctors. And Gemini is up to date with Google search and indexed information from different websites. But AI Studio just uses the default trained model. They do not have anything above that or beyond that. So Nived got some data as May 22nd, 2025. That means that model was trained on that day with the information having only that particular day. Until that day, they have all the information. Beyond that, the model does not know any information. Gemini is automatically the control settings are hidden, where in Google AI Studio, you can fully adjust how creative the AI should be, how much it should hallucinate, it shouldn't hallucinate, how it should be real or more creative or imaginative. And the AI Studio is more raw, highly creative and unpredictable. But Gemini is being trained or pre-trained and fine-tuned to be safe, constrained, as well as polite. So that's the biggest difference between using two different environments with the same AI model, right? So this is the reason why you're getting two different answers. And that's where I have to give you an overview about the foundations of AI, what are the different AI models and architectures, cross-functional bio AI domains, because we are talking about life, and also cognitive AI agent architectures, infrastructure, and local deployment. Let's see what are the current challenges and what is the requirement of AI. This is a reverse spiral where we have heard about Moore's law, right? But this is written as Eroom's law. This is basically the exponential gain in computing and drug discovery costs double every 9 years. This is a systematic inefficiency in traditional R&D. The R&D cost per drug exceeds 2.2 billion. The clinical trial failure rates are remaining stubbornly above 90%. That's where we are trying with bio AI to break this trend to predictive modeling, and target discovery as well as screening are being very much automated. So we wanted to see how we can implement this. And also the complexity barrier. It's there everywhere. Too much of data, too much dimensionalities of data. So how we can operate with multiple layers like genomics, proteomics, metabolomics, integrating these heterogeneous data types is the greatest hurdle in bio AI today. The integration, harmonization of these data is something very important that we have to look at. And that's where we have to look at how the foundations of AI can be brought into the picture to understand its architecture, mechanism, fundamental necessity of the modern machine intelligence. Today, I'm not going to talk about ChatGPT or Gemini, Claude, or whatsoever platforms that we have. I'm going to talk to you about the behind the scenes, especially what the foundation means, how we can tailor it according to our needs, and making AI to be your real assistant in science and research and in other areas. But why we need AI? It is mainly because of data explosion, which I told you earlier. You have data verticals and dimensions in multiple areas. And these multi-omic sequencing, real-time sensor streams, and spatial transcriptomics generate millions of data points per sample. Now, how can you synthesize them with high dimensional spaces with a non-linear over time? Uncharted sequence spaces also, universe of potential small molecules, protein folding, geometries larger than the number of atoms in the universe. So AI acts as an intelligent compass with high probability designs. And now let's see what is AI. I don't want to go for the old school talking about AI, ML, and statistics. I'm more looking at representing the design to model representation, recognize patterns, and extract features autonomously, right? When humans are very good at it, extracting and recognizing patterns and also model representations. But here we're looking at how to make use of the data that we have to understand the trend or the pattern, which is being optimized for mathematical objective functions and also complex sensory processing in silico. And this will be autonomous based on learning and adaptation. How AI can be applied in our real life or in our activities or tasks is one is predictive AI, the other is generative AI, and the third one is agentic AI or agentic automation. That's what is going on right now. We started with predictive AI, then with generative AI, it can generate all your images, or it can completely come up with new protein motifs or completely new structure based on reinforcement learning. But now we are talking about agentic AI. We'll see what is agentic AI in coming slides. Now let's try to compare between AI, ML, and statistics. Of course, without stat, there is no ML, and without stat and ML, there's no AI. So that's a foundation here. So looking at the core objectives, we are inferring the population and model relationship using statistics. We are trying to optimize the predictive accuracy using ML, and we are trying to simulate, generate, and act on these kinds of data inputs using AI. That's where we are looking at. And when you have a small enough of data, which explicit variables, we go with statistics. Medium to high, maybe hundreds, we go with ML related modeling. But ultra high, where it is multi-model unstructured, we always go for AI-based modeling. But there is always a bias. Statistics requires linear or normal distribution. ML assumes data bound but non-linear. But in AI, there's no prior structure. It's completely self-supervised. So to give some examples, in statistics, as you all know, students test ANOVA. And coming to ML, we have random forests, support vector machines. And for AI, we talk about transformers, diffusion, and multi-agents. So we have to have a clarity on what kind of data we have and what kind of method that we have to apply, whether it is statistics, ML, or AI. Now, in the due course, if you have any questions or if you have any comments, please put it in the chat. Once I finish my slides, I'll go through them. As I said, I won't be able to see the chat while I am presenting. Thank you. Now, let's see how AI learns to solve problems. First, we have to have the data. Of course, human collects the data, or they can also collect the data from the data which is available publicly. Train the model with billions of mathematical coefficients, minimizing the prediction errors through optimization algorithms. And finally, the predict the outcome, which is once tuned, the model processes the novel unseen text or input to predict the properties, generate text, images, or model physical environments. But for that, you need to understand the core components of AI architecture. One is the perception, what we talk about the computer vision or signal parsing systems that capture like microscopy slides, raw chromatograms, and physical bioreactor inputs. Looking at reasoning is cognitive graphs, semantic logic path that determine the causal links between the mutations and phenotypic attributes. There are databases for you to understand the genetic variations, including mutations related to some specific diseases or gene expression. But you need to know why, the reasoning, right? That's where this can help you with trying to harmonizing data from multiple sources of data that is available in different dimensionality. Now we take actions like hardware controllers or API calls that dispatches the molecular design to synthesize pipelines and coordinate plate readers. This is overall an automated system when we talk about action. But here, when we talk about the architecture and agentic AI, we are talking about reasoning, feedback, and iterations. Now, under the hood of AI, we are talking about tokens and embeddings. Generally, we learn back in school, computer science, right? Computers, when we learn, we talk about bits and binaries, right? So we talk about zeros and ones. The same way, we have to learn again what is AI under the hood. What is under the hood of AI? It is completely tokens, vocabularies, as well as high dimensional embeddings. Now, what are tokens? Well, tokens are set of characters. Let's say 4 characters can be called as one token. So before compute occurs, the text or DNA bases or chemical smile strings are being passed into standardized chunks called as tokens. So AI does not understand a word called as, let's say, biomanufacturing. They really don't know what that word is. The tokenization as well as the orchestration helps them to understand after bio manufacturing, there is a word coming next to it. So the AI can predict, the model can predict what is the text coming next to it. So these are the things will be covered in our further training programs where we have Python codes, which you can run it on Google Colab as well as on your local laptop, even though you don't have a GPU laptop, still it will run so that you will learn how these tokens are being used, what is the understanding of these tokens in our biological setup or even chemistry setup. Now, looking at embeddings, tokens are mapped into high-dimensional vector spaces where relative distance reflects semantic or biological similarities. For example, as I said, we need to understand after bio, there's a word called biomanufacturing or manufacturing. This is being given or inputted by a certain kind of vector databases or a lot of data that we give, sensible data that we give to the model in order to fine tune or pre-train them. So based on those embeddings, they try to understand what are we talking about. They can talk something similar to humans understand. So that's where the what we know about tokens and embeddings. We'll go bit more deeper into it during our more training programs. Now, looking at the foundation models and architecture, let's decode the neural network mechanics driving the biological sequence predictions. That's where the transformers come into place. Transformers are being replaced with recurrent architectures by computing relative dependencies across entire sequence simultaneously. So this is, for example, if you look at the left-hand side, the recurrent architecture is that it is a step-by-step process. They first take the data, they try to process it, and then at a hidden stage, and then they get the output out from that. So it's a long range. Dependencies are hard to capture. But in the transformer architecture, all at once are being processed. So you get a self-attention and it makes decisions by itself to position them in the sequence. So they get to know after bio, there's a word called manufacturing. So this captures a long range and global dependencies efficiently. So this captures the interactions, predicting the folding patterns or molecular binding profiles if you look at the proteomics area with high accuracy. Now, looking at the life cycle, when you look at any of the models, this is where you need to understand why AI Studio gave you a wrong date, but Gemini gave you the latest current date. The reason is pre-training and fine-tuning. Even though the models are being pre-trained in AI Studio, they were only for the data until May 2025 or May 2024. But they were being further tuned in Gemini to use the real-time data, which is from Google search or even from the existing live websites. So Gemini is able to give you responses on real time compared to the raw model, which was pre-trained earlier. So that is the life cycle here: self-supervised pre-training and downstream fine-tuning. So Gemini sits in the fine-tuning stage, and the AI studio sits in the pre-trained space. And that's why you see there's two different responses for the same question. Now, let's look into the biological context. Models that learn biological syntax by predicting masked elements in massive, unlabeled data sets, millions of protein sequences. They were not indexed or labeled, but they have builds for the general representation of chemistry, physics, and evolutionary conservation patterns. But when you try to fine tune it, we try to give weights based on the labeled target data. So we know which sequence is for which protein or genes or DNA. And then we will be able to focus on specific tasks like predicting binding affinity. This transfer learning reduces the need of large experimental dataset to train highly accurate predictive models. This is the reason why we always say that you need not to train or build a model from scratch. You already have foundation models where you can fine tune them or further train them. So fine tuning is basically you're trying to tell what is the question, what is the expected response, and you try to build that data manually and give it to the model and train that model. And you'll be able to achieve the accuracy. If you're getting 82% accuracy, that's really a great win. You can come up with a product, but very rare. You start with 50 or 60% of accuracy, right? That's the reason why in earlier days, when you try to ask number of letter R in strawberry, always the LLM gave a wrong number of R. But now if you ask what are the number of R's in the word strawberry, now it will be able to tell you properly. That fine tuning comes from human. The human tells to the model, these are the questions, these are the answers, learn it from there. That's where we have to look how these learnings and tuning can be looked at cross-domain bio AI application or cross-functional. Now, for that, let's understand what is bio AI. This is where the silicon meets the carbon, right? So BioAI is a specialized convergence of AI and life science methodologies focused on parsing biological sequences and molecular shapes. It can be translated into physical entities, computational codes that can be tested virtually. The paradigm shift transforms biology from an observational science to an engineering discipline. That's where we have to look at the scale of biology that we have. It's almost 3.2 billion base pairs are there in human genome, and which translates to more than 100,000 proteins and millions of molecular states and countless spatial, temporal, and interaction states. This is something that data beyond the human capacity. And this is the reason why we need AI tools or models to handle this kind of tremendous amount of data. So by treating the genomic pathway as an ultra large data set, the BioAI can scan millions of combinations to point disease causing genetic mutations in seconds instead of years. And this is where the target identification and target validation plays a vital role in recent days. So in my previous work in pharma, I was also working in target prioritization and target identification and extensively used AI models for that. BioAI in biology, again, proteomics. All of you have heard about AlphaFold, right? It has been a very great discovery on solving many of the protein structures. So they can map functional dynamics of the cell. Deep learning models predict intracellular protein complexes, phosphorylation states, and post-translation modification. As we all know, we need to understand the structure of the protein in order to define the functions, motifs, and domains. So that's where the scientists can target cell signaling pathways to understand disease and design therapeutic proteins. Now coming BioAI in chemistry, where we are trying to de novo design. By the way, all the images that I'm showing here, these are being generated by AI because since I'm giving an AI presentation, I should make use of AI also. So some of the images might not be real and most of them have references. So just letting you know that most of them are generated by AI. So AI explores a larger chemical space by representing small molecules as a graph neural network. And this is very important because recently the virtual chemical space is more than 32 billion chemical compounds. Can you imagine that can we synthesize that many compounds? No, but AI can generate it. So that's where we call it as exponential growth of the virtual space. The question is, can all this be synthesizable and really having application? The answer is no. But at least it was able to generate those many structures. Now, are they really meaningful or not? That is where humans here to filter them out, triage them, and get designed into bind to the target pockets in the proteins. There will be synthetic complexity also to these compounds. Now, bio AI in physics, where we talk about molecular dynamics, I'm just picking this as an example to accelerate quantum mechanical or molecular mechanical calculations. Machine learning models are already replacing slow AMD algorithms with neural network potentials. So in material science, especially, they are already using machine learning potentials because the physics-based algorithms for molecular dynamics, they take ages to finish because it's very computationally intensive, expensive, as well as for, let's say, for 10,000 atoms, it might take 3-4 days to complete the simulation. If I want to screen about 30 or 50 systems like that, it is going to take more than 3 months for me, including analysis. So in order to do an initial screening to triage the system, I would always go with a machine learning based potential, which is much faster. Of course, accuracy is a bit compromised there, but we have a bit more space to screen them. So we can screen thousands of systems and then come and end up with the three systems to do the physics-based algorithms. So your virtual screening will be much more feasible within the capacity of time as well as the computing power, so that ML potentials are really making much more impact in recent times in industry, because industry always looks at time and cost efficiency. Bio AI in genetics with high throughput. Of course, we have patient genome sequencing. Nowadays, we are talking about precision medicines, right? So I'm sure all the bio banks and data banks will be having all our sequence data with them, so that doctors will get access to these patient genome sequencing to have bit more understanding about what to do next or treatment. So we can understand the variant identification with the based upon deep learning prediction. Of course, it's evidence based. So interpreting clinical impact of rare genetic mutations are very, very important. So deep learning systems can help to look at identify pathogenic changes, how missense mutations can impact the protein structure. It's very important because mutations can make it specific, or even also can change how the disease has been diagnosed with or treated with. Now, looking at bio AI in fermentation, of course, you have real-time sensors in bioreactors. You have a data processing feature engineer. You could have some dependent factors or parameters for that, for example, temperature, pH, or dissolved oxygen. So based on that, we can try to understand the trend and also predict a control model out of it. And coming into biomanufacturing, you can look at a scaled industrial bioprocesses where we are streamlining the cell line design, downstream purification workflows, and this can also monitor the filtration system, purification columns to detect the fouling before issues occur. So optimizing the chromatographic cycling increases recovery rates of valuable monoclonal antibodies and biologics. So these are some of the representations just to give you an understanding how AI can learn the trends out from the data that we give. Now, bio AI in pharma with the drug discovery pipelines, we are trying to shorten preclinical development cycles. Recently, there was a drug that has gone to clinic, which took only 18 months in the preclinical studies. So that's where AI can help you to accelerate. Of course, not in every cases. It all depends upon how reliable to target active chemical structures, predict ADMET profiles, and identify repurposing opportunities. And these in silico models help pharmaceutical companies skip thousands of random assay screening, reducing R&D cost and timelines. But this is where the human in the loop is much more important here. The human has to assess whether these compounds or the predictions are sensible, amenable to make it forward, to make the decision whether to perform these random assay screening or not. And again, bio AI in microbiome analytics, where we look at the metagenomic classification, profiling complex microbial communities and their functional roles, where we can read the raw metagenomic sequences to identify bacterial species and predict metabolic output, and connecting microbiome dynamics to clinical health conditions like inflammatory disorders. Ultimately, our full health is completely dependent upon the gut health, right? And gut is technically the brain of our health. So we will be able to understand the synergy of our gut based upon the bacterial health or species to predict them. Again, bio AI in Ayurvedic system, where we have different formulation. Ayurveda is something very interesting. Back in 2017, 2018, when I was working with few formulations into understanding the network pharmacology, it was that each of the extract, let's say here you have ashwagandha. Ashwagandha itself has 60 different compounds. And then Pipali. Pipali will also have again 60 different compounds. It's not that easy for us to map targets for each of these compounds, as I've shown in this right side of the slide. So we need to understand the network mapping where AI can help us to predict these human targets for compound. Now, if you take that specific compound and if you try to administer, you won't get the same effect of the herbal formulation or Ayurvedic formulation. That's the composition that we need to understand. It's a combination of different compounds comes together for that particular mechanism and particular mode of action. And it's on multiple targets. So by mapping multiple compound interactions to biological targets, AI can translate traditional systems into evidence-based medicinal framework. But for that, what we need the right data, or we need to generate the right data in order to map these network. Coming to bio and nutraceutical designs, functional food formulation, right? Again, we have natural plant resources, which is diverse and also sustainable. So designing health promoting compounds or personalized nutritional plants where we come with peptide discovery. So our center is also expertise in synthetic biology under the leadership of Professor Dar. So there we are also looking at the peptide designs. So AI models can evaluate thousands of natural plant peptides also from the dark genome and then isolate functional ingredients, optimizing bioactive peptides to support gut health, lower blood pressure, and manage glycemic responses and few others. I just picked up few for the sake of discussion. But then looking at the prediction, we have to look at the multi-property evaluation, or we call it as MPO, multi-parameter optimization, where bioactivity, absorption, stability, safety, and allergenicity, these all has to be considered while we are going for screening. So, so far I was trying to talk about why we need AI and what is the components in AI, what are their architectures, what are the different verticals which can be used, how AI can help us in the bio AI segment. But now it's time to understand what are the agents, what are these cognitive agents that can help us. These are simply moving from simple models to autonomous scientific agents that reason, plan, and execute. Let me give you an example. Today, I'm asking Gemini, hey, give me a 1 day trip plan to Cochin, right? It will be able to give you some sites to visit, where to eat, the time to start, time to end, that's all. But I'm asking my agent now, hey, agent, please try to plan a trip which is within my budget and try to find out when my time is available away from my work, away from my other activities, and make sure I go at the right weather and it shouldn't be rainy. and make sure there are some festivals going around. So I have put a lot of conditions and requirements into my model. So it has to go to multiple sources. It has to check my calendar. It has to go to my office related work. It has to go to weather reporting. It has to go to festival reporting sites or something. And it has to come and make a decision. But each time it comes, okay, to my calendar next week, I'm fully busy. But next week, there is a festival happening. So it comes back, take the feedback, goes back again. Oh, no, it doesn't work because Giri is busy next week. Let me go back and make another decision to do or check some other week. So these kinds of iterative feedback system is called agent. They try to think themselves. They try to reason themselves, and then they plan, and finally they execute. So multiple areas are being covered or considered in order to give you a response, and that's where we call it as an agent. So what are AI agents? They are autonomous goal execution ones. And these are unlike traditional statistic models, which I already mentioned to you that these have used bit more brains in order to plan things based upon our requirement. And they try to connect with all possible resources and data to get the feedback. And they are also doing dynamic self-correction. Sorry to say this, recently we can see most of the people who were doing entry level coding, they have a tough time right these days because of the launch of Claude Code, as well as many others like Cursor, Lovable, and these many platforms are able to do coding. I'm I'm not from a computer science background, but then I was able to easily switch into quickly get into coding and scripting with the help of these agents very recently. Even though I used to code shell scripting very efficiently for many years. So that is where the code is able to self-correct themselves by trying to debug and understand what is error, what is a library. It has to be corrected based on the code crash, or maybe something, if you do a molecular docking error, it tries to understand where it went wrong, which parameter was wrong, and it will try to go back and correct it, and then redo the process again until we get the right one. But there is a hallucination factor here. It tries to see whether it is working only, whether it is amenable or meaningful for the next step, they don't check. Again, we have to make them learn doing that. I'm sure it will be capable in the next 6 or 8 months, but we need to have enough data to make them. But that is where human in the loop is coming into the picture. So the loop allows agents to handle complex workflows without requiring human intervention at every step. It's good, but we need to know what it does. So make sure human intervention is always there in each step. Otherwise, it can do anything messy. So we have to be very careful when agents are trying to do self-correction. Now, what is the anatomy of the agentic AI loop? As I told you, it looks like planning. They try to split into high level goals, different one, with a sequential task using reasoning technique, tree of thought or chain of thought. I already told you an example, like check Diddy's busy schedule from his calendar, check the festive time, check the climate, check the budget, and based on this, you have to plan a trip. I did not tell any day or anything. It has to give me a day which I can take it up, considering all my other goals or sequential tasks. And the tool, what are the tools that I should use? In my case, I've used Google Calendar, which is my calendar, and then maybe weather reporting sites and many others. But in biological sense, we look at blast results or Python scripts, which are already tailored for REST APIs or pulling data, molecular docking softwares like Autodoc or even Flare, and also literature search engines where we can use Google Scholar and many others. Again, coming to evaluation, because based on these tools, you also have to evaluate whether the planning that is made by the agent is viable or not. So the agent critiques to its own results against physical constraints and refine the design before outputting the data. So it asks the question itself. I think it's a good habit of asking ourselves also before telling the results whether it's really right, why this has happened, how this can happen, what will happen if I report this, these questions are being evaluated by itself by the agent, right? That's where it comes with the RAG system, RAG, or it is called as retrieval augmented generation. So now if you're asking, what is the effect of curcumin on inflammation? This is the question you're trying to ask. It tries to look at different journals, multiple journals, maybe peer-reviewed scientific databases. And then based on the LLM, it tries to generate a response. It tries to bring together all the summary, try to analyze by itself. Does it make any sense or can it connect? And then they try to do that with a retrieved context. So the LLM reads the retrieved messages and generate a response. Now, then they recheck back and see whether there is any evidence for this response that LLM has generated. So it goes back to the paper or the literature and tries to check, okay, LLM has generated this particular response. Does it map or does it claim the same thing that has been claimed in the paper or not? So this is verifiable references. That's where in initial days, when you were using different LLMs like ChatGPT or anything, you never got the real references. They cook up references. But right now, these days, at least today, if you try, you get the real literature references. So that's the beauty of RAG system. They try to learn themselves. So we are getting grounding AI into the scientific literature just because of the vector DB. All the papers in the PubMed or in European PMC, they all getting vectorized. These vectorized, once it is vectorized, only the tokens can understand. And then the models were trained based on that. So the models or your LLM can understand what is written on the paper, your research papers or publicly available papers. So they can extract key sections, append them to the prompt, and the model then generates a synthesized answer with the citations. And this is where it helps, really helps you to write papers. Please don't copy paste it, but at least it will help you to write papers. For example, in my previous job, we had to review more than 100 or 200 papers, but we don't have time to review that many. And we wanted to create a summary to make decisions. We have used kind of our RAG systems with this literature databases in order to make those decisions. Now, we are coming very closer to the end of this webinar. Now, we wanted to understand how all these things can be done, because I was mentioning about training models. Can we do it in Gemini? No. Can we do it in Claude? No. This has to be done offline. Because if you're trying to train some models with your data, with your expectation of outcome, you have to train offline. Or maybe in cloud, you can do it, but you have to pay for it. Not like Gemini or anything like that. So training and serving a secure, sovereign biological language models locally is something which is booming. And also many are focusing on that. The need of offline local bio AI is that intellectual property guarding. For example, at CVJ-Centre, we are focusing on offline local bio AI because of the intellectual property and also operational zero latency. These two are very key components that we are looking at. Because when we collaborate with industry, they also look at securing their IP. So proprietary lead compounds, drug design should not be sent over public APIs or preventing data leaks. So deploying local models ensures all structures, everything within a secure enterprise network. Operational zero latency, again, high throughput workflows cannot be dependent on external network or cloud server outages. So offline deployment provides consistent runtimes, which is essential. That's where we need to understand training LLMs on local hardware, how this can be. This we call it as quantitization, which is compressing 32-bit weights to 8-bit or 4-bit integers. And this allows large models to run on normal GPUs, even on your gaming GPUs. There are something called as LoRa adapters, which is low rank adaptation freeze, which base weightage trains smaller parameter layers, lowering the memory usage during the fine tuning, because these trainings are memory intensive. So in order to do that, we use LoRa adapters to get into smaller chunks of layers. And sovereign compute, as I said, to run open source model like Olama and then securely run them and deploy them. Now, what are the differences between on-premises and local cloud hosting? So we already have experience in localized sovereign deployments because data sovereignty is something very important. A risk of external training exposure, but here we are within the local firewalls, throughput controls, throttled by rate limits and predictable execution runtimes on the localized. And but bit more on a costing. If you look at variable pay per token API fees are available on cloud, but fixed upfront hardware investments are huge in localized sovereign deployments. Now let's look at the quantized local LLMs. I know this is a bit advanced for many of you, but this is something that we are heading to. For the next 12 months, after 12 months or 8 months, you're going to hear a lot about local LLMs, as you now hear about AI agents and agentic AI. So you will have a local workstation with your GPU, CPU, and memory and fast storage. And you can do agentic tasks on local LLM for data analysis, literature review, experimental design, code and simulation, and reporting agent. These can be fully automated so that you are much more around 8.4 times you can speed up. These numbers are all based upon the recent studies and reports. Now let's look at the real world translation and validation. A few empirical benchmarks in biopharma. So if you look at the data flow, we are having heterogeneous life science data sources like genomics data, proteomics data like mass spec and others, chemical libraries, and scientific literature data. We tried to do an orchestration layer in order to merge and relate whatever possible with the mass spec data, with your smiles and literature all getting connected. And then we have an agentic AI for reasoning and planning, where we are setting some tools in order to make that hypothesis. And then it goes to the knowledge augmentation data, which is called as vector database, which I told you already, where we have literature data or any other database, let it be anything. So then the RAG makes the decision whether there is evidence-based responses that is being created from the LLM. Then we go to the tools, which is from the pre-trained models like AlphaFold or DiffDog or MolMim or POMSE. These are all different tools used for different property predictions. And at the end, we come to the validation and discovery output, what are the possible novel target discovery for certain diseases, what are the optimized molecular lead molecules which is having lesser synthesis time, and patient stratification markers, which is clinically validated, where we have already explored patented data, clinical trial data, as well as published data. So based on this, we were able to come up with a discovery output. That's where we wanted to look at a self-driving lab workflow. Get get the data from experimental data or public data, go into the AI models, design the molecules or design peptides or any other parameters, rank them based upon prioritization. Then we give instructions for automated executions. These are all looked at the DBTL cycle, where we are looking at learn and then again design again. But the regulatory report generation is something very important when you look at more of compliance perspective. So you need to have an autonomous semantic matching hours, agentic documentation generation, automated and localized LLM auditing, and secure local compilation of draft. These are all very much required because of EHR screening, preclinical prep, compliance check, and drafting for IND. Now comes data governance and ethics. It's very, very important. You cannot use simply someone's data since it is publicly available. You need to know what is the license for those data or the right to use of those data. Anything, even for normal AI or even for, especially for bio AI. Because when you're going for bio AI, there might be patient data and health data. Privacy and consent is something very, very important. And then comes with the data security. You're talking about medical data. Security is the biggest concern there for biomedical data. Data quality and integrity. It's all about accuracy, provenance, and reproducibility. Then comes with the regulatory compliances like HIPAA, GDPR, bioethics guidelines. And then, very, very important, it's all on human responsible use, prevent misuse in research and clinical applications. This is where we need to have accountability and transparency on the data that we are going to use for these AI models. So please make sure that the data that you're going to use for any models is going through data governance and ethics. Now let's do a normal general comparison between bio AI versus manual. For example, our AI-driven virtual first, we start with data, AI models, then we do some virtual screening, select some candidates, and then validate in lab. So we start with thousands of virtual experiments, and we end up with 10 or 100 per week physical wet lab experiments. But when you come to manual or traditional way, and still the traditional way is the best way I would recommend for the time being, because the first one requires tremendous amount of data. So we need to only through traditional way we can generate that many data. So you can do literature search, hypothesis design, lab experiment, analyze, and then iterate. But here you do weekly experiments 10 to 100, but you cannot do virtual experiments, which we have done hundred thousands of compounds or sample data. We cannot do them. And the outcome will be just one. So we did not do a high throughput screening in order to make a decision. This is our final outcome. Rather, we always bias. Should we run a few more experiments to get better than this outcome? So that bias can be overcome with any of these virtual experiments. So you can scale up your virtual experiments, commit resources for the best candidates, as well as you can also limit it by physical experiments time and resources. So that is where you have to see you can screen molecules per hour around 10,000 using AI model, but using manual assay, you can do only 12 molecules per hour, maybe 96 based on the well plates. And on a structure prediction, AlphaFold takes only 3 seconds to predict a particular protein structure based on the sequence. But when you go for crystallography, it's weeks. Of course, accuracy levels are different, but again, if AlphaFold is able to predict, if able to find those structures within their training set, they are accurate. Apart from the fact about the stereochemistry issues and few others. So integrating AI models yield massive throughput gains, letting research labs scale up their virtual experiments before committing physical resources. Now, let's look at the future of the bio AI, which is like emerging friend, foundation model, synthetic biologies, as well as quantum scale integration. As I said, we look forward to a roadmap where we are looking at offline LLMs, which is completely locally deployable, and self-driving labs where we can synthesize some compounds, number of within 17 days, where from starting till end, which is a closed loop, everything is done by AI or robotics or digital twin. But let's see what is happening with the roadmap. In 2026, it's more of foundational models integrating proteomics, transcriptomics, epigenetics into singular representation map, which is multi-omics. And then 2020, another 2 to 3 years, we are looking for fully autonomous laboratory campuses running design, synthesis, validation, and self-correction cycles. In fact, robotics is already there in place in labs. In my previous workplace, we had robotics for high throughput screening and also for others. And then organ simulation, high fidelity simulation of whole human organs run digital trials, lowering the reliance on animal testing. We are also, CVJ Center is also an expertise in it. We have Dr. Sukanya, who is working on 3D in vitro models. So she'll be the best person to talk a bit more about it. She will be also having a webinar soon about that. And then looking at quantum scale at 2035, we are already started with the Quantum Valley and many more other initiatives. But then the quantum native deep models, we are still in the development stage. We have to calculate absolute atomic binding kinetics in real time and streamlining drug discovery or biological application, chemistry or physics related applications. So this is where the roadmap looks like. I'm sure these things will happen even before the years that I've mentioned in my slide. Now, I wanted to conclude my just the presentation with AI will not replace you or the labs because human is required in the loop. So when AI model generates something, human judgment is required whether this can be put in real life or not. Then you need a bit more improved decisions, and then you change your data, tweak your data, and the AI model learns it again. So you need humans for this. Again, coming to lab, even though there are AI models there, but you still need further parameterized, or from your experience, what are the parameters to be changed to have a better product, lesser impurity on your experiments. That data can be put into use to AI models to learn. So you have to make sure all the scientific guys, human in the loop and lab in the loop is something very important in your throughout your AI modeling life. Because make sure in any workflow, human should be there, and any wet lab related data or digital twin, as I'm working also on digital twin, lab should be there in the loop. We cannot avoid them. Now, that's all about the AI. I want to just talk a bit about the center. So, CVJ Center for Synthetic Biology and Biomanufacturing, we are a pioneering non-profit autonomous research center, as Pawan already mentioned, and we are engineering biology through AI-driven synthetic biology and biomanufacturing. We also have research and consultancy collaborations with Manus Bio US, NCH, Think Bio, NUS, and Synthite Industries. We also launched a first of its kind MTech program in synthetic biology and manufacturing in partnership with QSAT. So every year, new admissions happens. Even this year, also new people are coming in. We do have scholarship also for this MTech program. We also having establishing a 10,000 square feet advanced facility featuring genomic, metabolic, and process engineering labs, which is also equipped with the cutting edge bioreactors and fermenters. We are also driving industry through commercial consultancy, executive training programs, and translational research. Looking at the research portfolios under CSB, we do have 3D in vitro biology, bio AI, metabolic engineering by Dr. Shivangi, nano bioengineering by Dr. Srija, dark genome by Dr. Pavan, and functional genomics by Dr. Uni. So this is our team. We have a great, strong scientific advisory board and also a research team in different divisions and segments with a great expertise of industry, as well as many more experience in real life experiments. That's all from me for today. Thank you so much for your patience. As I said, we can have a discussion. This is more of an open session right now. If you have any questions, please put it in the chat, or you can request for unmute and I'll be able to unmute yourself. Thank you.
Thank you very much, Dr. Pillai. This was really enlightening. You took us from the very basics of AI, and not only that, but also its limitations and the scenarios where AI can be used in applications. And so I've got two quick questions before others get started, if you're okay with that. Yeah?
Yes, please.
Okay. So the first one is, as an educator, I'd like to know, for someone entering bio AI today, what skills are most important for turning AI ideas into actual products, whether they are medicine or biomanufacturing? So the skills that one needs coming from the biology community. So which skills would be most important?
Yeah, the first and foremost skill that is required for any student or any scholars or scientists coming to bio AI is the understanding of how this data is generated, number one. Number two, understanding about the foundational statistics, because AI can be trained with anything, but we need to know what is a parameterization that we have to give to get them trained. Rest of the infrastructure, others and all, you need not to know. There are standard facilities and infrastructures available for you. But the domain knowledge is something very, very important. So when you're coming from any domain, and if you're getting into bio AI, the domain expertise and statistics or machine learning methods is something that you have to focus on. These are the two take-home message.
Thank you. Thank you very much, Giri. So the second is as a scientist, my second question. Uh, can AI discover causal mechanisms that humans have not yet recognized, then merely identifying correlations? Is it
So
possible?
there's a great debate going between causality as well as correlation, right? So it is always a fight between statistics and real life. So sometimes in some cases it is possible, but not always. That is the reason why I always told you in my repeatedly in all my slides, humans should be in the loop for the judgment, because the causality mostly only human brains can act accordingly. But in certain cases, for example, numbers or economics or finance, trend, weather, these we may be able to predict or correlate well with the causality, but not on other science related, which is more dynamic, which changes every day based upon when you start from a random seed. So at this point, the answer is no.
Thank you very much. Yeah, I think it's very important for humans and AI technology to work with each other as colleagues
Correct.
rather than, go in the different directions. So thank you very much.
Absolutely. Absolutely. Thank you, Pawan, for those questions. Everybody, thank you all. If you have any questions, you can unmute yourself. The mics and the cameras are enabled. So please feel free. Or if you want, you can put it in the chat. And if you want a bit more think about the questions, you can always contact me, or you can send an email to this connect@cvjsinbio.org. So the email reaches to me, I can reply back with the answers if you think so. Otherwise, we are on time with the webinar, but I can stay for a few more minutes if you have any questions. Oh, hi, Kyle Viri. Very much. We need mathematicians for modeling, right? I hope you remember all your integrations and differentiation, which you were doing back, right? Including graph theory and numerical methods. These are very much required for AI modeling from scratch. Very much a mathematics researcher can get into bio AI modeling. Thanks for that question. Any further questions? Oh, by the way, we will be having advanced training on how to train a model, how to get into fine tuning these models. These are our executive training programs. We will be launching in the due course, how to run a local LLM. So much more into designing compounds or proteins, all things that you can do offline. So you will be understanding the science behind these models. Please email to connect@cvjsinbio.org. We will be able to get back to you, Kyle Viri. Thank you. Any further questions? Yes, Nived. I'm a student in BioAI. What approaches should I go for in future for researchers and work? Nived, it's completely, as you can see, I was explaining about different cross domains, or I call it as cross functions for BioAI in different verticals or different areas of domain, right? So you can pick one of them from there because it's also what you wanted to do next. It's because the market is so dynamic, you cannot see what is happening now and take a decision, right? You try to see, as Pawan always say, try to find answers for the unanswered questions, right? So that is the way that you have to look forward. Try to see what is being unsolved. What is that yet to be finding an answer for that question, and try to work for that. You might not go with an end result, but through the path, you will find out where you have to work or focus on. So my recommendation would be one of the domain that I have discussed. If that is of interest to you, you can pick them and see whether that really fits in with your interest and passion. Both is required. Thanks, Nived. If you want, you can unmute yourself if you want to speak up, or yeah, I'll go through the chat. Okay, I'll wait for 1 more minute and then we can end the session if you have any more questions. And please visit us. We are located within the CUSAT Kalamasheri campus, a beautiful campus. So please visit us if you're coming to Cochin. Always welcome to the center. Okay. Yeah, looks like there is not much more questions, but if you have any questions, please email to this email address connect@cvjsinbio.org. We'll be happy to respond to you in any means. And thank you so much. Thanks, Pawan, for the introduction and kind words, and looking forward. And thanks each and every participant who have been patiently listening to this. I hope this was an eye opener as well as a lot of insights being given to you, and we can discuss further. Thank you so much and wishing you a lovely evening. Bye.